Hi, my name is Jessica Anderson, and I'm going to be talking to you about CLN1 disease and the enteric nervous system. Now, I'm sure you've heard of neither of these things before, but don't worry, I'm going to walk you through them. All right, so CLN1 disease is a fatal disorder caused by an autosomal recessive loss of function mutation in the PPT1 gene. PPT1 encodes a lysosomal enzyme, and as a result, deficiency in PPT1 causes a buildup of storage material in cells. The central nervous system is particularly impacted, leading to neurodegeneration and early death. Multiple gene therapies have been attempted to replace the missing enzyme in the CNS, but CLN1 mice still die prematurely. We think that the enteric nervous system is the missing piece of the puzzle. New preliminary data suggest CLN1 disease also causes profound pathological changes in the enteric nervous system and bowel motility abnormalities. Gut dysfunction is also really common in children with CLN1 disease. So here we characterize and measure ex vivo neurogenic motility patterns in the colon and the distal small bowel of PPT1 knockout mice at an advanced disease stage, using something called an organ bath apparatus. And this brings us to the bottom left of the poster. So in the bottom left, you will see a diagram of our organ bath apparatus, which is essentially just a high-tech chamber in which we can take the bowel out of the mouse and keep it alive. It's pretty amazing. While the bowel is alive outside of the mouse, we can take videos of it as it's doing its thing. All of the things that it would normally be doing if it were inside the mouse, and that includes contracting. So when we take videos of these contractions, we can convert them into spatiotemporal maps called chymographs, and these maps show us when the bowels are having what's called a neurogenic propagating motility event. So a motility event that is traveling proximally to distally down the bowel that's mediated through enteric neurons, that is, the neurons that are in the bowel. And what we can see is when these events occur, how long they occur, and how far they travel. So what we've done in our first results section is we've quantified the number of CMMCs, and these are colonic motor migrating complexes. These are the neurogenic or neurally mediated contraction patterns that occur in the colon that propagate contents proximally to distally down the bowel. And so comparing the number of these in the wild type compared to the knockout, we don't see any significant difference. We also don't see a significant difference in the rate at which these events occur or the distance at which they travel. And that's all in the colon. However, when we look at the neurally mediated contraction events in the distal small bowel, we see that there's actually a significant difference. And that significant difference is specific to male PPT1 knockout mice. Therefore, our findings suggest that disease progression in the enteric nervous system is sex-dependent. Our findings also demonstrate a region-specific motility defect. This is really interesting because this study is the first to assess ENS involvement in pediatric lysosomal storage disease. And our findings are the first reported motility defect in an animal model of CLN1 disease. As you can see, our findings are really unique, and I'd love for you to take the opportunity to ask me questions about what this means for the field. Our next steps is that we're going to increase the number of mice evaluated, but we hope to eventually evaluate the ability of ENS-directed gene therapy to return normal contraction patterns to these mice. Thank you to Robert Hoykeroth, Bob, my mentor, for his many years of mentor and sponsorship. And thank you also to Jonathan Cooper, PhD, WashU. He brought us this project and this amazing bit of science. Thank you also to the CURF office for awarding me the College Alumni grant that has allowed me to do this work. Again, please ask questions. I'm thrilled to hear them. I really look forward to it. Thank you.